Everyone, I'm and it's my channel Pure Biology, and welcome to my channel with another video. I have already started Lac Operon and in my previous videos. So before following this topic, you first check out another important topic, what is actual Lac Operon and what is the positive and negative control and what are enzymes are related to the Lac Operon. You first check out, and I obviously give the link in the I buttons, or you can follow the playlist. Okay. And before going to the topic, I want to say thank you to all of you who have already subscribed to my channel and give support to me to grow my channel. Okay. And I will try to give different informations about the different topics, and that will be very helpful to all of you. Okay. So today's topic actually is the Lac Operon mutation. So actually Lac Operon, why? Because the lactose or inducer is very important here. And before going to this topic, I explain just briefly few words that there are two kinds of genes you can see. One are the structural genes and other is the regulator gene. So, and here the most important thing is the regulator gene, obviously, because its product is the repressor protein that is very important for continuing the transcription process or not. And here I will discuss about the mutation or some about the obviously regulator gene. If the generally regulator gene remain in wild type situation, then it is I plus. But when it becomes mutation. Sorry for the disturbance. If the gene is mutated, that means the regulator gene or the I genes that becomes mutated, in that situation, what condition is arising? Today we will learn. Okay. So regulator gene, actually when the mutation is seen, the mutated gene is like that, I minus. Okay, mutated gene I minus and I is another I also describe. I will describe that ID. Okay, ID. So first you understand or we will learn that Lac I minus. So in natural cases, what happened? The Lac I or the regulator gene, ignore the disturbances. Okay, just follow me. What happened in case of I minus? Actually I minus is the mutation of the regulator gene. Okay, generally I plus in what situation arise? It produces the regulator protein that binds to the operator site and stop the proceeding of the RNA polymerase and also stop the transcription translation process. But what happened in case of Lac I minus? In case of Lac I minus, they do not synthesize the repressor or inactive repressor that cannot bind to the operator. Okay, two situations may be arise. One, if the synthesis of the repressor is not possible, or sometimes as usually repressor is synthesized but they are inactive. Okay, so here this is one kind of example, that I plus O plus Z minus Y plus, and here I minus O plus Z plus Y minus. So what happens here? I minus that cannot bind to O plus, because here the regulator protein that is produced, that is inactive, or maybe the regulator protein is not synthesized. So, but in case of I plus, you can see here the normal repressor protein that will be formed. Okay, normal repressor protein formed, and this protein is sufficient for binding the two operator. Okay, don't needed this inactive repressor, because I plus is present. That means the regulator protein obviously produced, and that are very sufficient for binding with the two operator and to stop the transcription process. Okay, and in that situation, what happened if the inducer present? So the transcription is possible. Okay, but if the inducer absent, okay, inducer is absent, so in that situation the total transcription is not possible. And what situation arise in number one this and number two this? Here Z minus that produce the beta galactosidase that is defective, okay, defective. And here Y plus, that is normal permease. You can see that Z minus enzyme, that is defective beta galactosidase, and Y plus, that is normal permease. And in the second situation, the normal permease and the defective beta galactosidase. Permease is number 2. You can see the beta galactosidase normal. I have written wrong, I think so. Beta, this is not normal. This is defective. In cell number 2, the defective is permease, okay? Defective. This is defective, okay? Defective. And this is normal, okay? This is normal, okay? So what we learn here, that if I minus is present, okay? So the repressor protein that will form is either inactive or the repressor protein that will be not formed. But suppose this situation is example, is this type of gene or this type of situation arise, and what happened in that situation? I minus, you can see this is mutant, okay? So here the regulator protein is not formed, or if it is formed, that is inactive. But in case of I plus, there the regulator protein is produced, obviously sufficient, that block the 2O plus and totally stop the transcription process in absence of inducer. Clear? In absence of inducer. This is I minus mutation. The second one is the IS mutation. This is about, obviously the mutation is seen in the regulator gene. This S means the super repressor. This is called the super repressor, okay? This is very dominant, and if this one is present, so that means there is no need of regulator protein, because, or repressor protein, normal repressor protein, because this is sufficient, okay? This is very sufficient for binding the operator, okay? This is because super repressor, and this is dominant, obviously, okay? This is dominant, and there is another important thing is, this can no longer binding to the operator site, okay? Wild type, in case of wild type repressor, they can bind longer period of time with the operator, but Lac IS super repressor cannot bind longer period of time with the operator system. So here the trans-acting non-inducible. Non-inducible means it cannot recognize any induce, okay? Any inducer, so it cannot recognize any type of inducer, no lactose, no allolactose, no not other thing. So it's very easy to bind with the operator site. If inducer present, that means lactose present, or the lactose present, no effect is created on that, okay, because it cannot identify the inducer's presence or absent. So it can bind with the operator site, and as a result, no production of the Lac enzymes, and obviously translation is not possible. Suppose this is enzyme, their I plus and IS is very important. This is the situation, I plus and IS. So here the I plus effect, its effect is not necessary as IS is present. Why IS is present? Because it is super repressor, it is dominant, so it can bind to the operator site, both operator site, okay? Suppose there is O plus, obviously there is O plus. So it can bind both the operator site, that means its regulator protein, that is the super repressor, that bind to the both operator site, and there is no need of normal repressor protein that is produced from the I plus in the media. So here the actual transcription is not possible, and obviously the translation is not possible. This is the mutations of the I minus and IS. Another mutation of I, that is the ID. Now I will explain you. Okay. This is Lac ID minus, okay? This is Lac ID minus. D means dominant and minus is negative, okay? And here I produces the homotetramer, okay? Generally monomers can associate at random in the cell to form the active protein. Okay, in case of formation of the regulator protein. So monomer is very important. But here what we will see that homotetramer. Okay, here the normal and the defective subunit produced. Okay, that means within the regulator protein or repressor protein, that is non-functional. Okay, in case of the if ID minus that is present, so in that situation the non-functional or constitutive, because here the repressor protein or the regulator protein that is produced, but this is non-functional, not the functional, because some defective subunit also present here. With the normal, some defective subunit also present here, and that's why it can bind to the operator, but the operator is not effective, okay, or not affected by the binding of this. Okay, sorry. So in that situation, what happened? The transcription process that becomes constitutive. Clear? So this is example one diploid, that is ID minus and I plus. You can see the I plus that is positive or wild type, and in case of I plus that produce obviously normal repression. Okay, and this normal repressor, if the normal repressor present within the medium, what happens? So obviously that blocks the transcription process. But here, in that situation, defective polypeptide present in the repressor. Clear? As these produce the normal repressor, but these produce also the defective polypeptide. Clear? This produce the normal repressor protein, but this produce the defective polypeptide that consisting of repressor. Okay. So normal repressor is not found here. Okay. So here absence or near absence of the functional repressor, that means the normal repressor that actually help to bind the operator system to stop the transcription process, here that is produced, but due to presence of ID minus, the normal repressor is not found. So absence, you can see, or you can you can say that absence of repressor or near absence of repressor. Functional, obviously, the functional repressor here absent or nearly absent. So in that situation, what happened? The constitutive enzymes phenotype result. Clear? Constitutive enzyme phenotype result, because the D is the dominant. Okay, this is the D is the dominant. So what happened last? We can understand that constitutive enzyme phenotype result. Clear? So this is the ID mutation, ID minus, that is the dominant negative mutation. So these are the mutations today. We learned actually the mutations that arise in the regulator gene or the production of the repressor protein. Clear? So how the mutation of the regulator gene that affects on the production of the functional repressor protein, it is seen here or it is explained here. Okay, I think you can properly understand about this repressor protein or the formation of the repressor protein and how the medium is affected or not. You can properly understand. And in my next video, I will explain you about the mutation of the operator system. Okay, what type of mutation is seen in the operator system and the promoter system. And I think if you can understand it properly and you actually get some information about the Lac mutations, so obviously you must subscribe my channel and share with your friends. Okay, so I will discuss about the O mutation in my next video. Please continue or follow with my channel, and obviously you follow my Instagram. The description, in the description box I have already given my Instagram link, and I sometimes upload any video in that site. Okay, so please continue with me.